All right, so I'll be introducing Dr. Earl Miller. So Dr. Earl Miller is the Picower Professor of Neuroscience at the Massachusetts Institute of Technology. He has academic appointments at the Picower Institute for Learning and Memory and the Department of Brain and Cognitive Sciences at MIT. Professor Miller earned his bachelor's from Kent State University and completed his master's and PhD at Princeton University. Professor Miller is a pioneer in investigating the role of neural oscillations in cognition, and especially in the role of prefrontal cortex in executive function, including working memory, attention, and rule learning. His work critically combines experimental investigation, novel theoretical frameworks, and computational models to understand how neural oscillations arise from the collective action of neurons and underlie our cognition. Professor Miller's work has inspired many others to replicate and extend his findings. On a personal note, my first grad school experiment was doing just that. So it's a true honor to be able to introduce him to you all. I know you'll find his talk highly insightful, so enjoy. All right, Earl, if you're there, take it away. Okay, we cannot hear you. All right, we think you might be muted, Earl. Do you want to try that?
Yeah, how's that?
Oh, that's perfect. All right, go for it.
Okay, sorry about that. So I'll back up a bit. So today's talk will be about mounting evidence that cognition and consciousness arise from emergent property of the brain, its rhythms. I'll primarily be talking about cognition and consciousness as a related topic, and at the end I'll talk about the neural correlates of unconsciousness. But it wasn't that long ago that we had a very different view of the brain that didn't involve emergent properties or dynamics or even rhythms. And so I'm going to start with a bit of history to explain how neuroscience evolved to this view, because to understand the role of neural dynamics in cognition and consciousness, it's important to understand the kind of problems they solve for the brain. So now let's get in the time machine and go back to the 20th century when I was a graduate student. In the 20th century, we thought that neurons all had one function. Every brain cell, every neuron in your brain had one thing. Did one thing and one thing only. When it gave off electrical impulses, it was giving one specific message. And we also thought that anatomy was destiny. The key to brain processing is the pattern of wiring between neurons, almost like the brain was an elaborate telegraph system. And here's kind of an example of what I'm talking about. We have here on the right, we have a image on our retina of a little complex object. And the idea was there was a set of neurons that were selective for particular features of that scene, like edge detectors. It detected little line segments. It broke down the visual world into little line segments. And then those edge detectors combined into longer edge detectors that detected longer edges. Then those neurons combined their activity by virtue of this wiring to corner detectors, and so on and so forth, until we had neurons that had complex shape detectors. So you saw this process where the brain was thought to break down everything into component features, and then combine those features by virtue of anatomy to eventually lead to complex percepts. And you have lots and lots of neurons in the brain, so the idea was with this, you combine, and eventually you'll get peace, love, and understanding. Now, why do we think this way? Well, one big reason was technology limited us to studying the brain one piece at a time. Back when I was a graduate student, we had slow computers. We only could study the brain, the electrophysiology of the brain, one neuron at a time. And also importantly, we needed to understand the brain's pieces before we could begin to put them together. We can't jump in by studying how the whole brain works together. We first have to catalog its various parts. But now we think about the brain more in terms of network properties and dynamics. This is because we gain knowledge about the brain's parts. We understand what the individual parts do a little better. And new technology has allowed studying the brain in a more holistic fashion. We're no longer limited to studying the brain one neuron at a time. We can image the activity of hundreds or even thousands of neurons simultaneously. And that's a whole new ballgame. It has allowed us to have a better understanding of the brain on its network level, its emergent properties. And I'll get to an example of what those emergent properties are in a moment. And this has led to new discoveries about cognition and consciousness. Discoveries about cognition and consciousness that would not have been possible by studying the brain piece by individual parts. They only emerge at the level of these emergent properties. And this came about, as most things in science do, because the classic approach, the idea of thinking about the brain as clockwork, where every neuron has one function, and telegraph where the wiring is what matters, that couldn't explain new discoveries about the brain. And one critical insight was that neurons are not like gears in a clock. They're multifunctional. So as I mentioned, modern neuroscience, we're talking about mid to late 20th century, began by studying the brain's sensory inputs and motor outputs. They're the most obvious things to study. If you want to study how the brain perceives the color red, you look for neurons in the brain, in your visual cortex, that activate to the color red. And on the other end, the output was true for motor outputs. You want to study how the brain moves a lever, you study the motor commands in motor cortex that are involved in generating those commands. And again, we only studied the brain, these electrical impulses, from spikes we call them, from individual neurons, one piece at a time. Neurons give off things called spikes, these little like Morse code-like electrical impulses that play a big role in signaling one another. And this led to what I just explained earlier, and that is this idea that the brain works by these specialized neurons that are wired together in particular ways. And here's an illustration here on the right of this idea of traditional neural coding, this way we used to think about the brain. In this case, not edge detectors. Now we have neurons that are selected for things like form, color, and motion. And they may combine to produce a percept of a few related features like form, color, and motion. Red plus form plus motion equals a percept of a red rolling ball. And that's the way we used to think the brain worked. But newer technology, the ability to study many neurons simultaneously and get a better feeling for what whole populations of neurons are doing, not individual neurons. And also, our knowledge allowed us to ask deeper questions, not about sensory inputs and motor outputs, but about cognition, the middle of the brain, deeper questions about how the brain works. And that led to the discovery of neurons that were multifunctional. They didn't have the traditional neural coding we thought about in the 20th century. Instead, these neurons showed nonlinear mixed selectivity that combined many different feature combinations. They no longer showed selectivity for or activation by single features or combinations of a few related features. They combine every possible feature you can think of in many different ways, and they do so in a very nonlinear, unpredictable fashion. And importantly, these neurons also adapt their signaling to play different roles in different situations. In other words, neurons in cortex didn't just do one thing. They were like utility players on a baseball team, or jack of all trades. They could change what they do from moment to moment depending on the cognitive demands needed at the time. And they had no obvious combinatory principle. It wasn't like color, form, and motion add together into a red rolling ball on the level of individual neurons. Brain activity, neural properties seem to be way more complex than that. And when you see things in the brain like nonlinearity, the first thing you should think about is complexity, because nonlinearity breeds complexity. So what about these neurons, these multifunctional neurons that are so important for high-level cognition, the deep questions in the middle of the brain? Well, my colleague Stefano Fusi and Matti Ragati, working with Melissa Warden in my lab, created a mathematical model, a computational model of the brain that showed exactly that. They showed this property of nonlinear mixed selectivity adds computational horsepower to the brain. They knit together a wide range of information. They make the brain smarter by creating a high-dimensional representational space where more complex information can be represented. They also increase the brain's storage capacity by distributing information among many neurons. The brain could store an almost unlimited amount of information. And this multifunctionality allows faster learning and cognitive flexibility, which is exactly the properties we associate with high-level thought. The bottom line is that what they showed in their computational modeling is you can't build a smart brain without multifunctional neurons. Well, okay, but this multifunctional neurons was different from the old way of thinking about the brain, but they also raise another challenge for the old way of thinking about the brain. It's not just the wiring. Multifunctional neurons means anatomy in the brain is not destiny. Instead, it means that neurons are members of multiple networks with changing participation. It's not just how the neurons are wired up. There's some other organizational principle that allows neurons that are members of different networks to selectively participate in one network or another. So how does the brain organize? If it's not how the brain is wired together, not the telegraph system or the clockwork of how the neurons connect to one another, how does the brain organize its own processing? And this is a critical question because cognition is the brain organizing and controlling its own processing. Simple creatures can just react to the environment. They can swim towards food, they can run away from predators, they can avoid light. This is the environment organizing brain processing. But higher order animals like us, we can organize and control our own thoughts. We can choose what to pay attention to. We can make decisions. We can emit voluntary actions. We can pursue goals. And this is all about the brain organizing and controlling its own processing. How does the brain do this? Well, that insight came from examining the brain at a higher level, its emergent properties, which is the subject of today's talk. Now, what are emergent properties? Emergent properties are not explainable by individual parts alone. Like, for example, here on the lower right here, we see a video of a crowd doing the wave. And hopefully you can see the wave moving from right to left across the computer screen. Well, the wave in a sports stadium is an emergent property because you can't see the wave by studying each person separately, even if you study the whole crowd. If we approach this crowd the way we used to study the brain back in the 20th century by studying the brain one neuron at a time, I could, for example, give a microphone to everybody in this crowd one at a time. And I would have very detailed information about what each person in that crowd is saying, but I would never see the wave. The wave only comes about by seeing their real-time interactions in real time, see how they interact, how the different component parts, the neurons, the people, interact together. And your brain has an emergent property. The most obvious emergent property of your brain are neural rhythms. Your brain oscillates. It oscillates anywhere from 1 time a second up to 30 times a second or more in a bunch of different frequency bands, all nested inside one another. Your brain is very harmonic, constantly producing these rhythms that nest inside one another, and harmonize inside one another. And these neural rhythms reflect the organization of millions of neurons. They reflect millions of neurons activating together, then deactivating together in a coordinated fashion, much like this crowd doing the wave. And it wasn't that long ago that these brain waves were dismissed as being like the humming of a car engine, an epiphenomenon, because they just didn't fit into this idea of neurons having one function and everything in the brain was wiring alone. It was thought that these are just the fumes given off by the brain. But now we've learned much more about them. In fact, just as a simple stadium crowd can self-organize using a few simple rules, like stand up when the person on the left stands up and then sit back down again, evolution can harness these same dynamics for the brain to control its own processing, to organize its own thoughts. And just imagine the possibilities. This kind of scale of organization, we see the level of brain waves, millions of neurons coordinating their activity. This scale of organization is exactly what is needed for high-level cognition. Paying attention, making decisions, producing voluntary actions requires coordinating a large number of neurons around a particular function or goal. And that kind of large-scale coordination is exactly what brain rhythms provide us. They're a natural organic organization. My guess is that when nervous systems got complex enough, they naturally began oscillating, as any network of electrical elements will do. But once you have a simple network that's oscillating, that means neurons are activating together and deactivating together. That is organization. And evolution just found a way to push around those waves, to push around those oscillations, and allow the brain to control its own processing. And also, there's something called representational drift. If you study neurons one at a time, you realize that neurons are very fickle. They come and go. If you study the same group of neurons, or even a single neuron, 10 or 20 times in a row, they'll activate differently each of those times. Neurons will often do a burst of firing, then they'll be quiet for a while, then they'll fire again, then they'll be quiet for a while, then they'll fire again, they'll be quiet for a while. Neurons just come and go and only occasionally contribute to ongoing processing. So the cortex is like an orchestra of very fickle musicians. The song plays on, the cortex is producing function, but each member only contributes a snippet of the song. Musicians come in, they play a little bit of the song, then they're quiet for a while. Then they come back in, they play a little more of the song, then they're quiet for a while. The cortex is like an orchestra where musicians are constantly coming and going, each only playing a little snippet of a melody. So if you want to control the brain, it's much easier to control the brain on the level of these emergent dynamics instead of on the level of these fickle individual players, which not only are hopelessly complicated, it seems to me, but also unreliable. In fact, you can actually read the contents of thought, not from individual neurons. Well, you can also do it from individual neurons, but you can also read them from the brain's electric fields. And here's an example of a study by my colleague, Dimitrios Panosis at the University College London. And what we showed is that we can actually read the brain's electric fields, and we had a subject trained to hold different images in working memory, hold them in mind over a very short period of time. And what we found is that we could actually read the contents, read what image the subject was holding in mind from the brain's electric fields. Now you can do this by the level of individual neurons, but it requires a lot of averaging over time, over multiple repetitions, because the neurons themselves the individual neurons are very fickle. But by looking at the brain's electric fields, you can actually read these thoughts in real time, and there's no representational drift. It's just a pure signal. Now that sounds like something very useful to me. Now it's not just useful for us to study the brain, it's also useful for the brain itself, because these electrical fields have a causative influence in the brain through a process called affective coupling. The idea here is that synchronous oscillatory firing of individual neurons create localized electric fields, and these electric fields then turn around and train neural activity via this process of affective coupling. Basic electromagnetic principles are a two-way street. The neurons create these electric fields, and electric fields turn around and influence the neurons. And this is, to our mind, a great way for the brain to begin to organize itself, to create these electric fields and then move the electric fields around to control what the processing the brain is doing. Now, not only does the brain, we think, use electric fields to control the brain on the macro level on the order of millions of neurons, we also think it influences the brain on the micro level. And here's another study from Dimitrios Panosis, where we hypothesize, marshaling a considerable amount of evidence, that the electric fields can restructure the brain, again, not only on the macro level, but on the micro level, on the molecular level, fine-tuning networks to make them oscillate and process information more efficiently. So the way to think about this analogy is if you put a bunch of grains of sand on a speaker and you play a note through the speaker, the grains of sand will form shapes. And this is the idea. These fluctuating electric fields can actually move around the structural proteins that make the brain's cytoskeleton, that produce the structure of the brain. And what they do is they fine-tune the brain, like fine-tune the networks, like fine-tuning a guitar string, like tuning a guitar string, to allow the brain to oscillate, produce these rhythms more efficiently. And because these rhythms, because these brain waves are carrying information, it's a way for the information the brain is processing to restructure the brain on the molecular level, literally a mind to molecules hypothesis. So the way to think about this is brain anatomy is not destiny, like we used to think. Brain anatomy is more like the road and highway system. It just says where traffic could go. This road and highway system is very important. It is the basic information, the structure, the long-term memories of your brain, but it's not thought itself. Thoughts is how the traffic runs on the road and highway system. And traffic can run different ways on this road and highway system. And we think the brain's rhythms, these oscillatory dynamics, these moving brain waves, these oscillating brain waves actually direct the traffic. In fact, we've found evidence that your brain kind of operates like FM radio. It uses different frequency bands to carry different types of signals. Now here here's a human brain. We have the back of the brain is where your brain's sensory cortex arises. This is where sensory information is processed. And the front of the brain, especially the prefrontal cortex, is the executive part of the brain, that one that produces the control that allows you to gain control of your own thoughts, produce voluntary action, choose what to think about, choose what to pay attention to. And our lab has been finding evidence that sensory information is carried by high frequency, above 30 hertz gamma rhythms. The sensory signals from the external world ride on these high frequency gamma rhythms, presumably coming from the back of the brain to the front of the brain, from sensory to executive cortex. And we found evidence that beta rhythms between 12 and 30 hertz, just below the gamma rhythms, they reflect acquired knowledge, not the outside world, but the knowledge the brain has acquired about how the world works, the rules of our society, the rules of the game. Whatever task your brain is engaging in, the knowledge of how to accomplish that task is carried by these beta rhythms. And we believe we have evidence that these beta rhythms are like control signals. And the way they produce control is they suppress gamma. Okay, so you have sense information from the external world flooding into your brain, and the beta rhythms act as a suppressor that sculpt and shape the information from the outside world. And this suppressive process that allows the brain to filter out unwanted information and pay attention and do all sorts of things that we associate with volitional control. It allows top-down control. That's what executive control is of brain processing. The focusing of attention, decision-making, voluntary actions, organizing thoughts and directing them towards goals. This push-pull between the external and the internal world, between the gamma rhythms carrying the external world and the beta rhythms controlling them. So the way to think about this, we incorporate this into a model which we call spatial computing. And the idea behind spatial computing is the brain is processing by using these lower frequency beta waves, kind of like a stencil. The beta waves target and selectively inhibit specific patches of your cortex, leaving untouched cortical networks that could be activated by sensory inputs or motor outputs, which are also carried by gamma waves, when needed. So in other words, your brain organizes thoughts by setting guardrails by these beta waves, and then let reality color in the details versus gamma waves that fill in between the lines. That's the idea. And in fact, so mention these gamma rhythms are carrying information about the external world, from sensory cortex to frontal cortex, whereas the beta rhythms, these control signals, this acquired knowledge about the world, is carrying them in the opposite direction. So to explore this further, we studied the cortexes layers. The cortex has 6 layers, kind of a 6-layer onion. And the superficial layers of the cortex, the ones most on the outside of your cortex, they are the ones anatomically that carry feed-forward projections from your sensory cortex to your frontal cortex. Those are the superficial layers. Whereas the deep layers, we know from anatomy, carry signals in the opposite direction, feedback projections. Whoops, typo there, should be feedback projections from the frontal cortex back to sensory cortex. So we looked a little more deeply at the rhythms within these layers, and we indeed found evidence to support our hypothesis. Namely, we found evidence for a circuit in cortex between these layers that generates and allows the interplay between these beta and gamma rhythms. We found that the beta rhythms that are carrying the top-down control signals are, as expected, stronger in deeper layer cortex, the feedback layers of cortex that carry signals from your frontal cortex to your sensory cortex. And they indirectly, by coupling to beta rhythms in your superficial cortex, to weaker beta rhythms in your superficial cortex, they suppress and control the gamma carrying bottom-up sensory signals from sensory cortex to your front of your brain. So the push-pull between deep layer beta control signals and superficial layer gamma sensory signals is what allows your brain to gain control over its processing and allows you to maintain this tone where you can process the information as needed, but not allow the external world to overwhelm your brain. Now, here's the most data-driven part of the talk. We found that this circuit motif, this beta in the deep layers of cortex that are carrying the feedback signals, and the gamma in the superficial layers of cortex that are carrying the sensory signals, we found this literally everywhere we looked in cortex. So this was shown here. Here's recordings. So this is 7 different cortical areas. We actually studied 14 different cortical areas in this study. And what it shows is, just to simplify things, here are the alpha beta rhythms in blue. Alpha beta rhythms in blue, the gamma rhythms in red, and this is strength on the x-axis. This is weaker is on the left, stronger is on the right. And on the y-axis are the different layers of cortex. 0 being the middle layer that separates the superficial layers from the deep layers. Literally everywhere we looked, we see at the beta rhythms, and a little bit of alpha too. It dips down a little bit into alpha below 10 hertz. But these lower frequency alpha beta rhythms are stronger in the deep layers of cortex, whereas the gamma rhythms are stronger in the superficial layers of cortex. And we found that literally everywhere we looked at cortex. We also found it in different species of primates, macaques and marmosets, in the human brain and also in the mouse brain. So it's literally this motif, this circuit motif, whereas lower frequency rhythms control high frequency rhythms, beta controlling gamma, we find it everywhere we look. And it's so robust, you can even find where that middle layer of cortex is in cortex by finding the crossover point between these rhythms. And when you see an effect that is this ubiquitous and robust, you know it's doing something very important, something fundamental to what the cortex does. Now, so far I've treated these rhythms as if they're like standing waves, like a jump rope that just oscillate up and down in place. But it's much more complex and precise than that. These rhythms form traveling waves. They don't just jump up and down, stand in place like a jump rope. That actually wouldn't be very useful for brain function because it would mean that whole networks would activate and deactivate. And we don't want whole networks activating and deactivating. We want precise surgical control over activation in networks. And that's what traveling waves do. This is an example right on the left here of a recording from the lateral prefrontal cortex of a subject. It'd be on the side of my head, side of your head too. It's the part of the executive part of the brain, right on the side of your head, right above your eyes. And what each square shows is where we had a recording electrode from an electrode array. It's measuring, the array was formed in a grid. And we're measuring electrical potentials. We're measuring voltages from the electrode array. And the color represents the voltage. Red means more voltage, blue means less voltage. And what you can see is the voltages, the electrical signals are not activating the whole network up and down, this whole array of activity up and down. Rather, the electrical potentials move and rotate around the surface of cortex. In this case, it's rotating in this counterclockwise fashion. Now, why traveling waves? Why are traveling waves so important for brain function? Why are they so important for cognition? Well, they give very precise control over cortex, as I mentioned, because the waves can move across cortex and sculpt in activity wherever it needs to be. But also, traveling waves are super helpful because they buffer information about time and recent network activity, which are the nuts and bolts of what your brain needs to do computation. And the best way to illustrate that is you can take a video of a pond and drop in 4 pebbles in a pond one at a time, and just by taking a still photograph of that pond, you can look at it and you can determine where the pebbles dropped, how long ago they dropped. Right? So this is just the existence of traveling waves alone provide the brain with information that is super, super useful for doing computation. But also, these traveling waves are highly organized. They are not just ping pong balls bouncing around your brain randomly, bouncing from one to another. They're actually highly organized, just like those fans in the sports stadium. So this is a lateral prefrontal cortex, again, on the side of my head or side of your head of a subject performing a working memory task. And the posterior, or in the back of the brain, is in this direction. The front of the brain is this direction. This is the top of the brain and the bottom of the brain. And what we see is why the subject is performing this working memory task, holding an image in mind. There's 2 principal directions of travel of these waves. They move back and forth across the surface of cortex. One direction and sweeping back in the other direction. Just constantly sweeping back and forth across the surface of cortex. Much like that sports crowd doing the wave in a stadium. So when you see organization in the brain, you should think function. Because when the brain organizes something, whether it's a map of the retina or a map of your body for somatosensory information, organization means function. So not only are they highly organized, they change direction with cognitive demands. So now this is a plot here. There's a plot here on the y-axis. Sorry about that. Plot here on the y-axis of the direction, these 2 directions of traveling of movement. And on the x-axis, there's my cursor. On the x-axis is time. And what's going on in this diagram here is during this baseline period, the subject is waiting to start the working memory task. And here's where we give the subject an image to hold in mind, which it holds in mind over this memory delay, and then it makes its response. Which image did I see? So the important thing here is the task is beginning here and it ends here. And what we see at the very beginning, the waves are moving in both directions back and forth across the surface of cortex. And the moment the task starts, one direction drops out and the other direction takes over. Now it becomes very unilateral, moving in one direction across the cortex until the very end of the trial when the task is over, and now it goes back to this default state of the waves moving in 2 directions. So we know these waves are highly organized and they change direction with cognitive demands. Well, so what do? Well, we found very recent evidence that these traveling waves of electrical fields actually correspond with shifting patterns of spiking of the electrical impulses of individual neurons. Again, spikes are the little impulses that individual neurons give off, whereas these traveling waves are manifest in the electric fields generated by the spiking. And what's shown here, in the middle of this graph here, this is a representation of spiking activity in cortex using something called subspace coding. What subspace coding is, if I record the spiking from 1,000 neurons simultaneously, I can create a 1,000-dimensional space that explains what those neurons are doing. It's self-evident. Then you ask the question, how many dimensions, I have 1,000 neurons and 1,000 dimensions, how many dimensions do I actually need to explain what these neurons are doing? And the answer is all you need is 3 or 4 dimensions. All you need is 3 or 4 dimensions to describe what these neurons are doing. And what that means is that spiking patterns in cortex, at least in the mathematical sense, are not this impossibly complicated, well, it's going to be complicated, but it's not this impossibly complicated pattern of spiking of neurons doing these things that's impossible to decipher. Rather, what this means is that neural spiking, these individual impulses given off by individual neurons, is highly coordinated. Like birds flocking together across the sky. They change and they move together. That's what individual neurons in your cortex are doing. They're coordinating their impulses, their spiking, like birds moving together across the sky. And we have found direct evidence that these shifting patterns of spiking of individual neurons directly corresponds to these moving waves of electric fields traveling across your cortex. When we see rotations in this pattern activity of spiking, when they rotate around in this mathematical coordinated space, we see waves of electrical activity rotating around the surface of cortex. Now again, this subspace code and these spiking patterns, they're a mathematical abstraction. There was no a priori reason to think that movements like rotations in cortex, in these spiking patterns, would correspond to actual movements across the geometry of the surface of the cortex, but we found that there's a direct correspondence. And it raises the possibility that computation is somehow mapped out in this electric field, in this dynamic space in the brain's electric fields, and the way the brain does computation is by moving electric fields through that computational map. And because of a factor coupling, because these electric fields have a causative influence, that means these traveling waves of electrical influences aren't just reflecting these spiking patterns from neurons in cortex. They can organize neural activity. They can provide the means to create the kind of coordination we see in this flocking activity of individual neurons in the brain, this coordinated spiking from individual neurons. Now, why would your brain want to use these electric fields? Well, as I've been mentioning, it's a way of organizing neural activity. But also, electric fields have a property that is very advantageous for the brain. Synaptic transmission, the sending of impulses down neural fibers like axons, and diffusion of chemistry across the chemicals across the synapse, it's pretty slow. But electric fields propagate influences nearly instantaneously, at nearly the speed of light. It seems to be propagating influences, having this crosstalk of these waves moving between the wires and sending signals back and forth nearly instantaneously seems to be a pretty good way to organize brain processing. Now, this leads us to the topic of loss of consciousness. It may be disturbing to know, but medical doctors don't really know or haven't really known why general anesthesia causes your brain to lose consciousness. And there's been this tacit assumption that it simply just shuts off your cortex. Your neurons just stop spiking, activity goes away. So we began a series of investigations with my colleague Emery Brown at MIT, who's also a practicing anesthesiologist. We began a series of investigations where we went to look at what happens in your brain as you lose consciousness due to general anesthesia. So here's what your brain looks like in the awake state. These are recordings of electrical potentials. These are labeled LFPs. They stand for local field potentials, but they're recording of electrical fields on this more macro level. And here's the individual impulses given by individual neurons in the brain. And these are 4 different cortical areas, 2 at the front of the brain, frontal cortex, and 2 at the back of the brain, parietal cortex, auditory cortex, for you anatomy fans. And what we see in the awake state is a lot of high frequency radio chatter. Networks are constantly forming and unforming, and these rhythms are nesting inside one another because your brain is forming these networks, it's moving waves around, it's controlling, processing, a lot of chatter going on. But then when you administer to the subject a common anesthetic, propofol, you see that all this high frequency chatter that we've been associated with cognition is now replaced by a low frequency 1 hertz hum that swamps out and eliminates all the high frequency oscillations, the rhythms that we associate with cognition. So, but here's the mystery. Your brain also does this when you sleep. When you go to sleep at night, not when you're dreaming, but during what's called slow wave sleep, your brain shows a very similar pattern of low frequency delta 1 hertz hum. But sleep is very different from anesthesia. You know, when you sleep for a long time, you wake up feeling time has passed. And also, sleep is very good for sleep is important for memory consolidation. Sleep helps you solidify memories in your brain. What your experiences from the day, what you've learned from the day, gets solidified at night when you're sleeping. Memory consolidation. But general anesthesia, by contrast, causes amnesia. You not only lose all passage of time when you're anesthetized, but you lose the memories for the few minutes before you actually got the anesthetic. So why is that? Well, you kind of see a hint of it here. It's not that the brain shifts into this low frequency 1 hertz hum. These signals get kind of misaligned and offset from one another. So, and that's what's different between anesthesia and sleep. Is not that the brain shifts to another frequency domain. All the signals become scrambled or misaligned. And here's a recent study showing exactly that. This shows that after the subject was anesthetized, using in this case 2 different anesthetics, you get it scrambles the alignments of these low frequency waves. All the waves below 30 hertz, including 1 hertz and above, all the ones that we associate with this control processing in the brain, the beta and below, they become misaligned. They go out of phase with one another on one side of the brain. Then across the 2 hemispheres, they become more aligned, which is very different from the awake state. So it isn't that your brain shifts to a new frequency domain. The signals, the brain waves become misaligned from one another. And that prevents brain areas from talking to one another, and that presumably prevents things like experience of passage of time or memory consolidation because the waves no longer reinforce one another. Now, we did this study using 2 different anesthetics, anesthetic doses of ketamine and anesthetic doses of dexmedetomidine. Now, what's interesting about that is that these 2 different anesthetics act on different receptors in your brain, in different parts of your brain, different neurons, different parts of your brain. Yet, despite these different molecular actions on different neurons, these 2 different drugs alter brainwave alignment in the same way. If I would show you these data for these 2 drugs, you wouldn't be able to tell which drug is producing this misalignment. It looks identical across the 2 drugs. I want to emphasize this point because, in other words, the changes in brain waves can explain a phenomenon, the convergent effects of different anesthetics that can't be explained on a more reductionistic level, right? If these different drugs, we also have evidence that propofol does the same thing. These different anesthetics, they act on different parts of your brain, different molecules, different receptors, but they do the same thing. They make you unconscious. How do you explain that? Well, my more reductionistic colleagues who are focused on the brain's individual parts, and sort of, to this day, there's a few of them who still deny this emergent level. They can't, in principle, explain why different anesthetics all produce the same effect, because they're not acting on the same neurons and the same receptors. But we can, and this suggests that consciousness can't be explained on a more reductionistic level. It can only be explained at this emergent level, where we see these common effects that alter consciousness and cognition. So that leads us to our punchline that consciousness and cognition is an emergent property of brain dynamics. We think of it used to be thought that spiking in anatomy was everything, but now we kind of think of spiking in the brain, these impulses that individual neurons give off. I kind of think of them as the buzzing in the mouthpiece of a trumpet. The notes start there, but the real magic happens in these resonance patterns of the sound waves in the body of the trumpet. So anatomy, this detailed intricate anatomy in your cortex, for example, what it is it's a very beautifully exquisite, complex set of trumpet mouthpieces that produce these electric field properties, these dynamic electric fields, and that's where a lot of the stuff happens when it comes to cognition and consciousness, not at the more reductionistic level. So in other words, neural activity underlying, and in summary, neural activity underlying cognition is not smooth or steady state like we used to think. It's complex and dynamic. There are emergent properties like rhythms and traveling waves that organize neural activity, and self-organization of neural activity is the essence of cognition. This organization is central to cognition because the human brain, I think we can control and organize our own thoughts. And in a similar fashion, the emergent dynamics can knit together our conscious experience. Now, I know I spent much of my time talking about cognition and not consciousness per se, but it strikes me that many major theories of consciousness involve knitting together networks across the brain so we can have our unified experience of all the sights, sounds, feelings, and knowledge and thoughts in our head that create a unified conscious experience. And for the same reason these dynamics can organize thoughts, organize brain processing, in a similar fashion, they can knit together the unified experience of consciousness. And we found that loss of consciousness is associated with the dramatic alternations in these brainwave dynamics in a way that it can explain the different effects of different anesthetic drugs, which can never be explained on the more reductionistic level. So, I thank you for your attention, and I especially thank all the students and my postdocs in my lab who do all the hard work in creating the science. I'm happy to take any questions you might have. Thank you.
Earl, hello, this is Stuart Hammeroff. Thanks for accepting my invitation. And it was a great talk. I do want to respond to one point about reductionist ideas not working in terms of anesthetic mechanism. All the anesthetics that follow Meyer-Overton correlation indicate a unitary action, that they're all acting in the same way. And while they may bind to different receptors on the membrane surface, they all bind to microtubules inside the neuron, which accounts for a reductionist and unitary approach to anesthetic action and consciousness. And most of my anesthesia research colleagues disagree with me, including your friend Emery Brown, I would say. But I'd be happy to debate and discuss and take this concept further, because I think it's very important. Your comment, please.
Sure, yeah. I mean, you just offered a combined reductionistic and emergent explanation for the effects of general anesthesia, which is fine. I mean, that's our goal, right? We're going for that. Sorry, I for some reason I can't turn on my video. It says host is preventing me, so you can't see me, but I can see you. So I think, oh, there we go. So Stuart, so I think the kind of explanation you're talking about are the things that unifies different levels. They produce these reductionistic effects, and the reductionistic effects then lead to this emergent dynamic, and that can explain the common effects of anesthesia. So I don't think there's any incompatibility between what I'm saying and what you're saying. You're welcome to debate with Emery if you like, but I think we're kind of on the same page. I'm dealing with the signals that I can measure, which are these gross electrical signals from the brain, these electric fields. I can't see inside the microtubules, but given that I think that a lot of cognition and brain processing is rhythmic in nature, it wouldn't surprise me if there's some involvement there.
All right, thanks a lot. We still can't see it, but you're sure handsome in the picture we have. See you in September, I hope.
Absolutely.
Yeah, as you sort of summarize the dynamics and the waves, they're kind of essential for the brain or the consciousness. If I use the terminology from computer science, are these like waves and dynamics, are they necessary or are they sufficient for the consciousness? In the sense, if somebody simulates these same patterns and other things using other devices, this is not coming from brain, then in that case, will you call that consciousness?
Hey, everybody. Yeah, I mean, we know that endogenous electric fields, for example, will entrain neurons. So we know that they're having an effect, but your question is whether they're necessary and sufficient. I would say they are necessary. Sufficient, no, you need the neurons too. You need both the reductionistic level and this emergent level, both. So I would say that they're necessary for consciousness, for cognition, but they're not sufficient in and of themselves. You need the trumpet mouthpieces to create the emergent patterns of waves that produce cognition.
Thank you.
I hope that answered your question.
Yeah, thank you. Yeah.
Hi, thank you for a great talk. I was wondering if the traveling waves you haven't really talked about excitatory versus inhibitory. Do you think they can be both, or like in the circuit motif, the ping motif, but do you know that you mentioned? Yep. It's all about inhibition, driving the cortical waves. Do you think that happens at local level, or the waves can be inhibitory as well?
The waves are both excitatory and inhibitory, right?
Okay.
And this EI excitatory inhibitory balance is how the waves get created. And there's work by Nancy Capel and others modeling this, where the way you get different frequencies of brain waves is you shift this EI balance, this excitatory inhibitory balance. And when there's more excitation, you get higher frequency waves. Less excitation, you get lower frequency waves. And for that reason, when we first came across this phenomenon of the beta-gamma push-pull, where beta seems to inhibit gamma, where low frequencies inhibit high frequencies, we thought about it more in terms of energy state, that it's not a matter of inhibition per se, but what it is that if you drive the brain, the cortex, down into lower frequencies, that's a lower energy state, so you'll get less neural activity, less spiking. And if you drive it to a high energy state, you'll get more spiking, and that's where you get gamma, the higher frequencies. But now we have some new evidence suggesting that the beta waves, the beta band particularly, they activate inhibitory neurons in your brain directly. So we think there may be actually a direct inhibitory influence. So when you think about it, the gamma waves are reflecting high levels of excitability, and when beta wave comes around, it inhibits that excitability. And that's how the brain sculpts itself.
Okay, thanks. I have many more questions, but I'll leave to others.
Thank you very much, Eric. A wonderful, very clear presentation, and I'm sold on the brain waves. And thank you for your clarification that it seems to be you were focusing more on the cognition than consciousness and some of its issues. But I suppose the model begs the question, if these brain waves are organizing neural activity for cognition, where is the information, even the impetus, for the waves themselves? Where does that come from? Where does that reside?
Yeah, where's the homunculus? Right? Well, you know, what the brain does is represent information. Okay, so the control must come from a representation of the goals and the means to achieve it. Whatever task you have in hand, your brain can activate the sensory signals, the cues in the environment, activate a template for how you act in a restaurant, right? So it's got to be a representational issue, okay? And we think what happens is that there's these closed loops between the prefrontal cortex and the basal ganglia. And when you see closed anatomical loop, imagine a snake eating its own tail. And when you see that in the brain, and you see that between the prefrontal cortex and basal ganglia, what you should think is recursive processing. Those circuits are gated by dopamine, which is your brain's reward signal. So the reward, positive feedback, good things happening, sculpt this recursive process. And what it does is it creates representations that stamp in whatever led to the good outcome. And then through this recursion, you get this open-ended kind of nature where the system can learn something and then feed that information back in, that new knowledge back into the circuit for more elaboration. And then you get these representations that reflect this the task and the means to achieve it, the goal at hand, how to behave in a restaurant moment to moment, and that's where the beta waves come from. That's our hypothesis. You don't need homunculus, you don't need a little puppet master telling the brain what to do. What you need is information, and you need to express it in this way where it can get control over the rest of the cortex.
So recursive feedback loops generate the beta waves.
I would say the beta wave is a product of this recursive processing. Recursive processing generates a representation that's in that road and highway system of your brain, in the infrastructure of your brain, and then when activated in the right way, it then generates the beta waves. That's the idea.
Thank you.
Amazing talk, Earl. Thank you. Two-part question. First, to what degree does your approach invalidate or kind of transcend and include the spike code approach? And then second, to what degree are the traveling waves and the kind of macro structure that you talked about supervenient, but in a way almost independent causally from the spiking of the brain more generally?
Yeah, it's funny when the modern neuroscience, this emergent property sort of way of thinking about things is relatively new. And there's still a lot of my colleagues out there who think, oh, you're doing nothing but indexing and spiking, as if spiking is doing everything, and I'm just measuring it rather than with these brain waves, rather than actually looking at something that's functional. But that never made much sense to me, because spiking and electric field, they're the same phenomenon, just at different levels of analysis. Right? You can't separate the two. So I think it's artificial to say, oh, well, spikes are functional and oscillations not functional, or can you show me a function of waves that's separate from the spiking? You can't separate the two. It's a way. The waves are a way for the brain to organize. The spiking generates the waves, and the waves are a way to organize spiking. So you can't have, just like you can't have notes in a trumpet without buzzing your lips in the mouthpiece, you can't have the waves without the spiking, but then you can't organize the spiking without the waves. So it's all tangled together. It's not something that we could really separate in the brain.
One quick follow-up. So in terms of causal structure and effect on consciousness and cognition, you mentioned that it's much, much faster in terms of causal impact in the fields themselves. Given that far more granular spatiotemporal structure in the macro fields, isn't that the vast majority of causal structure in consciousness?
Well, I would say that the spiking act, the gamma, that's when you think about these waves, gamma high frequency, beta lower frequency. Frequency also means spatial extent of influence. Okay, so we think the gamma waves, we think they're the contents of thought, sensory information, because they're local ensembles, local networks that reflect what you're thinking about, the contents. And what the beta waves are is they're acting over a larger spatial influence because they're lower frequency. And I think the beta waves might be the right scale to produce organization in the brain that's functional and that can be used for cognition. Then you get to lower frequencies like theta and delta, which is like 4 to 8 hertz or 1 hertz. Now those are kind of state changes in the brain. Their spatial scale is too long to be really too wide of an extent to produce real organized function. So those beta waves are the right scale to produce, to sort of herd the cats of the contents of your thoughts in order to produce something that's functionally meaningful in the brain.
Thank you.
I hope that answered your question.
Mostly.
Mostly, okay.
Prior to your most excellent talk, my understanding was that if you used electrodes on the surface of the brain rather than through the skull, you would find standing waves on the surface of the brain that disappear every 10th of a second and then spontaneously reform. And that gives you a symbolic explanation, perhaps, for making the statistically most likely explanation for all of the inputs coming from other centers and creating a reality. Does that fit with what you've been saying?
Totally. There's a wide variety of waves in the brain. Now, I didn't mean to imply that they're all of them are traveling. There's both standing waves and traveling waves. But these traveling waves have a periodicity to them too, which in fact they rotate around the cortex. One very common motif in traveling waves are these rotating spirals of traveling waves that have a periodicity. So all this kind of fits with this idea of this periodic sampling. cognition is not smooth and steady state. For example, if you take a subject and have them do a sustained attention task, there's going to be a dim target that's going to appear on the computer screen. I want you to press a button whenever that target appears. And it's very hard to see. So the subject's really got to sustain their attention, pay attention the whole time. You think sustained attention would be the most steady state thing you can get in the brain. Well, you do that, it turns out the brain waxes and wanes about it around alpha theta rhythms. And your ability to detect that target waxes and wanes at the same rhythm in line with these waves oscillating in your brain. So even the most sustained thing, even the most steady state thing you should happen in the brain is actually happening in this periodic way that explains things like sampling of the world in a periodic fashion, not steady state sampling, which we now know the brain does.
Thank you.
Okay, awesome talk. Two things. So I was curious to hear your thoughts on the shift from component dominant to interaction dominant complex system theory as a way of talking about the mind and the brain. It seems like this is really compatible with what you're saying and could help with some of the sticky issues in terms of causation. And then two, have you considered calcium waves issued by astrocytes as synchronizing neuronal excitability? If not, I can send you some stuff I'm presenting on this.
No.
later today.
I'm totally glad you brought up astrocytes because
Okay.
that's one of the things I say to my colleagues when they say, "Oh, spiking is everything. It's a neuron spike. All this is just indexing spiking." Well, a good proportion, like 40%, a huge chunk of neurons in your brain are astrocytes.
Yes.
Astrocytes do not spike. They don't give off these little sharp impulses, but they do have membrane potentials that spread electrical influence. They're like neurons that don't spike.
Calcium
So a good
waves, yeah.
portion of neurons in your brain are spreading these electrical influences, these waves, and not spiking at all.
Right.
Okay, so
it seems like a second language, right?
yeah, well, not only that, but the way and often people will say to me something like, well, these membrane potentials in neurons are small and active potentials are big. How does one influence one another? Well, that's because your cortex, your brain is not a single neuron. Your brain's these network of neurons, right? And small effects across millions of neurons amplify. So what happens that, think about 80% of the time, or I'm making up a number here, but most of the time, neurons in your cortex are not spiking. They give a little burst of spiking, they're quiet for a while. Another little burst of spiking, they're quiet for a while. Even when they spike, they could potentially spike up to 500 hertz, but they actually spike more like 10 hertz. So most of the time, neurons in your cortex are not spiking. What they are doing this whole time they're silent is their membrane potentials are fluctuating. They're oscillating because of the fluctuating electric fields just below the threshold to produce a spike. Which means that most of the time, most of the neurons in your cortex are teetering like Humpty Dumpty on a wall. Am I going to spike? Am I not going to spike? Am I going to spike? I'm not going to spike. And all it takes is a small change in those electric fields to flip them one way or another. Right. That's the reality of how your cortex actually works. So when people say all these waves, these electric fields, they don't have an influence, I like you to explain me how they don't. Because what did evolution? What did it do? Did it evolve like some sort of Bose noise canceling mechanism to get rid of these influence? Because they must have an influence. It's just the way things must work. And plus, these oscillating electric fields are so strong that we can measure them outside the skull 150 years ago. And if evolution didn't find a way to take advantage of that strong signal your brain's giving off, I don't understand evolution.
Yeah, gotcha. Just one follow-up comment with astrocytes as well. They're interconnected through gap junctions to form a functional syncytium, enabling them to basically work as a collective unit. Thoughts on that as potentially addressing some of the things that you raised as well?
Oh, absolutely. Another way to spread electrical influence is independent of spiking and synaptic transmission. Okay. When I say this stuff, some of my colleagues say to me, why do you hate spiking? Why do you hate synaptic transmission? I don't. If there was a spiking neuron here, if there was a synapse there, I would hug it. Everything starts there. But saying there's something more going on at a higher level doesn't negate what's going on at the lower level. It all starts there. It's all very fundamental. So that's kind of my message to my colleagues, is that spiking anatomy and synapses are very, 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 very important. It's all very fundamental, but there's more going on. And what's wrong with that?
Yes. Amen.
Amen.
Hi, thank you for the talk. I have two simple questions. What happens if to our brain if there's a very high electric field around our head? Would it like ablate our consciousness or possibly enhance? And second is, what is the role of thalamus in your model?
Great question. Well, first of all, if you put a strong enough electric field in your brain, you can induce unconsciousness or all sorts of problems. But we know from a functional standpoint, for example, if I put a fluctuating electric field from outside the skull and I have it influence the cortex, at a level that's compatible with the cortex's own electric fields, you actually entrain neurons. So that's proof right there, or evidence right there, that the electric fields are having a function. The thalamus, yeah, that's really important. The cortex didn't just evolve out of nothingness, okay? The cortex is like an add-on to all the subcortical things that were there before the cortex came along. So you really can't understand the cortex without understanding the subcortex too. And the thalamus is a big, important contributor to these rhythms. Okay, there's evidence that the thalamus is what's providing these lower frequency control rhythms to the cortex that then the cortex then uses for control. But it may be happening by these loops between the cortex and thalamus, because there's long recurrent loops between the cortex and thalamus that seem to provide these lower frequencies like beta rhythms to the cortex. So we have to understand this is a system working together with the subcortex, generating these rhythms that then the cortex can then exploit.
Amazing, thank you.
So thank you for your talk. In the earlier slides, you mentioned that math has shown that multifunctional neurons makes the brain smarter. How do you define smart, and how did you reach this conclusion?
Well, the way that my colleagues, Stefano Fusi and Matteo Regata did this, they built a model using a standard network model with connectionism. And they built it two ways. They built it with neurons that all had neurons were all specialized and did one thing only. And then they started adding in these neurons with this multifunctionality property. And what happens is that it realizes that there's limits. You can measure how much information a network of neurons can process. So for example, if under the old way of thinking about things, if I have 1 million neurons, and those 1 million neurons all do the same thing in a highly coordinated fashion, you might as well just have one neuron. So you can actually measure from electrical activity, from the spiking activity, how much information that can be embedded in networks. And what they showed was that not only do these multifunctional neurons, first of all, they provide this high dimensional representational space. When you only have neurons do one thing and they act in a very linear way, like 2 spikes plus 2 spikes equals 4 spikes, then the brain can only solve very, very simple problems. But it's only when you have these multidimensional neurons, these multifunctional neurons, that produces high dimensional space that allows the brain to solve more complex problems because it's a higher dimensional representational space. But the other thing it does is that it allows the brain to distribute information across many, many neurons. So each neuron is contributing a little bit to the function instead of it being highly specialized in single neurons. So they showed mathematically, showed in computational modeling, that if you build a model the old way with specialized neurons, the computational model can only learn 1 or 2 or 3 or 4 simple tasks, then it saturates. And that model could have 1,000 neurons. You could add 10,000 neurons. It doesn't make a difference because you've already saturated the what specialized neurons can do. But you start adding in these multifunctional neurons, and all of a sudden you get all these benefits, including the brain not only solving more complex problems, but the brain goes capacity unlimited. It can learn as many tasks as you throw at it. And interestingly, across all of our studies, we found that these multifunctional neurons, when you sample higher cortex, they're about 30 to 40% of the neurons in cortex, higher cortex. And Stefano and Matteo found the same thing in their model. What made the model act optimally is when 30 to 40% of the neurons in this model were these multifunctional neurons.
Thank you.
All right, we have time for 2, maybe 3 questions. Yep.
Thank you very much for your presentation. Daniel Lappin with Intelligenesis Designs. So my question is about embodied cognition and systems biology. And so I appreciate the fascinating detail that you're presenting around the rhythmic structure of the brain. I'm curious of your approach and perspective on how brainwave rhythms relate to spinal, cerebral spinal rhythms, okay, going into like ultradian, circadian rhythms of the whole body, and looking at even endocrine system rhythms, such as very obviously a female menstrual cycle rhythms, and how that relates to the concept of cognition is potentially bigger than just the brain itself, where you look at cellular development in fetal development, in utero fetal development, prior to the development of the spine and the central nervous system in the fetal itself, you have a variety of molecular structures in a blastocyte, for example. And I'm curious, because that brings up the questions of, where is the center of computation, or what is the nature of computation, or the nature of attention across organ systems in the body, and the concept of volition across organ systems in the body, such as volitional mechanisms of the heart, for example, or the enteric nervous system. Is it a distributed model of cognition in the body? And so I'm just kind of curious of kind of where you see that, because certainly the brain itself is this phenomenal engine, but a car is more than an engine. For example, you got to have four wheels, you got to have doors, you got to have a steering wheel, like that. Kind of what's your approach, and kind of where do you think that trend is within the field?
Well, I don't have all the answers, and believe me, answering all those questions that you write a book on that. They're a fascinating set of questions. But I will say, this is going to sound glib, but it's not, is that the entire universe around you oscillates. Your body oscillates, your brain oscillates. It's not surprising to me that your brain would evolve this oscillatory dynamics to control its own processing. And yes, there's increasing evidence for brain-body interactions, and this is going to have a rhythmic component too. Breathing, heart rates, actually your brain entrains your respiratory rhythms and heart rates. So all this processing, both in the body and the brain, is all nested together in this harmony of rhythms, as is the whole universe. So it doesn't surprise me that so no doubt what you're saying is true, is that there's going to be distribution and of function, and there's going to be in training on multiple scales, not only inside your brain but to your body too. So there's just a lot left to be a lot of questions to be answered.
Awesome, thank you.
A question, let me see if I can language this. So you're saying the gamma waves, right, are carrying a mass amount of information, and then they get filtered by the beta. Is that correct, in essence?
Correct.
Okay. Have you seen, so we got inhibitory and we've got excitatory, right? That it can function in. Have you seen any, and the beta manages how we would act in a restaurant. So in essence, rules and roles of external reality. True?
Yep.
Okay.
Yes.
Have you seen any connection to language and meaning and symbol modulating that?
I haven't per se in my works, I don't study language, but I have read papers where people have talked about these beta-gamma interactions for all sorts of functions, including language. I don't keep my finger on the exact studies right now, but if I can take it out of language for a second, this anti-correlation between beta and gamma has really been well known for a long time. Early studies of motor cortex. You have someone like you say, press this button, but wait, don't press it yet. Wait till I give you the go signal. And the whole time they're waiting to press the button, you get lots of beta activation in your motor cortex, inhibition. Then the moment they get the go signal, the beta drops, and now gamma can be expressed, and the motor command happens. So even this is something that's happening. It happens all over cortex. This push-pull phenomenon where beta controls gamma. We see evidence for it all over cortex and for all sorts of functions. And I'm sure it's also going to be true for language too. I can't put my fingers on the studies, but I do know that I've seen a few studies where they talk about beta-gamma interactions in the language domain, but the details escape me, I'm sorry.
Got it. Yeah, because it seems like meaning regulates this awareness possibility that gamma has availability to pick up, but then meaning and purpose and symbol and, in essence, the memetics of the larger community, actually, that's what governs the ability to actually interact with this gamma on a high level.
Sure. And in line with that, we did a study a while back where we looked at subjects that make judgments about the exact appearance of images versus abstract representations of the images, right? And what you see is gammas engage when they're making literal judgments about what things actually look like, which is consistent with it being the contents of thought. But then when you have to abstract away from the exact appearance and think about them in the abstract sense, then beta takes over.
That's good. Thanks.
All right, we have one more minute if you have a fast question here.
Yeah, I'll try to keep it quick. I liked your talk and I like your energy, Earl. I have two quick questions. One is whether you're familiar with Michael Levin's work on bioelectricity and whether you see any relevance between that and your current research.
Sure, yeah, I've talked to Michael Levin about this, and what we talked about is how there's, in developmental biology, there's evidence that your body's cells migrate to a certain place because of gradients of electric fields. So how does a skin cell on one part of your finger know it's on this part of your finger and not on another part of your finger? And the idea is during development, there's this electric field gradient gets set up literally in the shape of a hand, and cells migrate along those electric fields to produce structure. And if your body does that when it's during early development, and your brain's this electrical system, it seems to make perfect sense that your brain would also use this to fine-tune the structure of your brain, which is the basis of our cytoelectric coupling hypothesis. I was talking to Michael about this in particular, and he found a lot of a lot of commonality between what we're saying about the brain fine-tuning itself with electric fields on the molecular level and the structural developmental biology.
Cool, thanks a lot. My second question is whether, under your current best understanding of how the brain works, whether it's possible for quantum fluctuations to chaotically amplify to yield big macroscopic effects, or whether you think they might get all washed away by thermal noise.
I have no way of answering that based on what I've measured. I'm certainly willing to entertain the possibility that these quantum fluctuations or rhythms are having a downward influence. Downward, I mean to lower frequencies that I can measure. I have no direct evidence for it, but I have no direct evidence against it either. And I think that again, I think if you err on the side of the brain nesting all these rhythms on multiple scales, I think you're probably going to be more closer to the truth than the old telegraph way we used to think about the brain.
Cool, thanks a lot, Earl.
All right, let's thank Dr. Miller for his talk one more time, and you're off for lunch.
Bye, everybody.